I keep on forgetting. Yesterday we spoke of two hormones being released by the stomach. You remember names? What one was gastrin, right? And what was it doing? It was communicating with something. What was it communicating? I am forgetting. Huh? What is the name of the HCl secreting cell again? I am forgetting. Parietal cell. Parietal cell. Good. And what was the other hormone? Ghrelin. Good. That sounds familiar. And that ghrelin is released by the cells, and the ghrelin. That's right. You are right. Ghrelin communicates with what? It's a hormone. It comes from the cells in the stomach, and it talks to what? Brain. What is hypothalamus? What does it talk to? Hypothalamus. And what is the message that is encoded by this peptidergic hormone called ghrelin? Hunger. Very good. Okay. And then that hunger in trigger the information that is issued by the hypothalamus will be now translate into motivating what kind of behavior? Food intake. Search for food. Okay, good. Now we will move on, and so the food is there in the stomach. It has received plenty of acid, and the pepsinogen and the pepsin, and the different proteins have been digested, or at least large molecules have been broken into small molecules. And then after say about half an hour or 1 hour of digestion in the stomach, okay, then the pyloric valve. Hello, pyloric valve, which is guarding the opening from the stomach into the duodenum, okay, which is closed. Okay, closed because of the latch formation. It stays closed, and suddenly when these now after the process has gone, now the valve will gently open. It does not open and keep open. It gently opens and closes, and as the stomach gives a strong peristaltic action, the valve opens and the food is actually ejected from the stomach into the duodenum. And in the duodenum again, you have now duodenum has to be very careful because it is receiving strong acid, okay, and duodenum is not as much equipped as the stomach to deal with so much of acidity. Therefore, the nature has provided that duodenum will have a rich secretion of bicarbonate. Okay. Now the beauty here is okay. I'll come to that. Let's take a look at this cell. What is this cell? Pancreas. Pancreas have lots of acini. That's exocrine pancreas. Let's forget about endocrine pancreas. Exocrine pancreas, the cells, acini cells, and they're actually responsible for the secretion of pancreatic enzymes. They have those zymogen granules. The zymogen granules are actually vesicles. In the vesicles, you have all the enzymes, okay, which are responsible for the digestion of fats and proteins and carbohydrates. And that's one of the richest source. Okay, and then you have enzymes like trypsin and chymotrypsin and carboxypolypeptide. All sorts of enzymes are there in the pancreas. And here we have, the author has given us what? One pancreatic cell. But then, like, you know, it's actually very much similar to the salivary gland. There are acini, and there's a duct, and there are hundreds of them. Okay, and then ducts all are connected. Okay, and then finally there's a common pancreatic duct. And then as the pancreatic duct opens into the duodenum, it is joined by the bile duct. Hello, are you with me? Okay, and the entire system is stimulated when the food arrives in the duodenum, and the duodenum will be flushed with what and what? With the with bile proper and the pancreatic juice. So we are talking of a single cell of the pancreas which is there to secrete the. So this is a zymogen granule. It is going to have different kinds of enzymes which are secreted by the exocrine pancreas. Good. On the basal side, we are having the different signaling molecules and its receptors which will excite the cells, and when it is excited, it will release zymogen granules and its contents. Are you okay so far? So look at this image system and tell me what are the three very important secretagogues. What word am I giving you? Secretagog. Say that again. Very funny word. Okay, almost sounds obnoxious, but there is nothing obnoxious about it. Secretagog. Okay, so I'll just frame a sentence. You will immediately get the meaning of that. These are the three secretagogues which will act. They are only hormones. They will act on via their receptors. The receptors are sitting on the pancreatic cell. Okay. And in response, so what are the three secretories? One is secretin. Now, where is secretin? Secretin is again yet another peptide hormone. Yet another what? Peptide hormone. It is secreted by what? It is secreted by the cells in the duodenum. Okay. Or I will say that just as we had two hormones in the stomach, ghrelin and gastrin, I am drawing your attention to the two hormones that are released by the duodenum. So, is duodenum endocrine gland? Yeah, duodenum is also an endocrine gland. And what are the two hormones? They are again peptides. And what are they actually? We heard them yesterday, I will talk about them once again. One of them is called as secretin. What do you call it as? Secretin. So, in the wall of the mucosa of the duodenum, there are some cells. Some people also like to call them as secretin cells. Does the name Starling mean something to you? Hello? You remember Frank Starling? Okay? Yes, yes, good. We remember that name. A great scientist, great physiologist. In the year 1905, just see how old, okay, he for the first time demonstrated the presence of and physiological significance of secretin. I am just telling you, opening in front of you an interesting page from the history of physiology. What is the name of the hormone? Secretin, okay. And what it did was cut all the nerves, okay, cut all the nerves to the duodenum. And when in the case of a dog, and even in the dog, when the food arrived from the stomach into the duodenum, the physiology kept on working as if nothing has happened. Then he said, if the nerve, if in spite of cutting away all the nerves, the things are still happening, there must be some communication. Okay, and then eventually they proved that this hormone, okay, and that hormone eventually happened to be what? Secretin, okay, and he is the one to coin the term hormone. You get it? History. Okay, good. Let's come to today now. Okay, so here we have the, okay, so the acidic food has arrived from the stomach into the duodenum. So the secretin cells which are there, they first experience sudden arrival of acidity, they get excited. So, who is exciting the secretin cells? Are you getting my language? The cells which they are sensitive to pH, and when the pH is low, they start secreting. So, and it's again G protein coupled system, ATP, adenyl cyclase, cyclic AMP, intracellular signaling cascade, and it will be released in the okay. Now, actually secretin, that hormone, has some action on these cells, but it has a more profound action, much more profound action on the duct. Hello, duct. So, there is an acynus, okay, a few hundred cells. They will secrete the pancreatic juice, and then there is a duct. Are you with me? And there are hundreds of ducts, and they all join. Finally, common pancreatic duct which will open into the duodenum. I am talking about the lining of that duct. Okay, and those lining of the duct, they have receptors, and they have receptors for secretin. And those cells, those lining the duct, they play a profound role in the synthesis of bicarbonate. Now, what you did there in the stomach, okay, now you have to do something opposite. Okay, so you have acidified, now you need to what? Neutralize. Okay, so whereas these cells of the proper pancreas, they secrete the enzymes which are present in the zymogen granules. The ducts do not have zymogen granules, though they do not secrete the enzymes proper, but then they secrete a large amount of water, okay, and a large amount of bicarbonate ions. Okay. And again, now just saying that bicarbonate hormones is okay, but more interesting is it has to secrete as much bicarbonate ions so as to neutralize the amount of acid, so that you ultimately end up with the pH of around 7.2 or something like that, which is characteristic of duodenum, not too high, not too low. So, just depending, so you have to, there is titration happening, okay, and that is being governed by, okay. We have already done it yesterday, I will tell you once again, there is yet another kind of cell, okay, and that cell is responsible for the secretion of CCK. Now, tell me the full form of CCK. Cholecytokinin, perfect, cholecytokinin, right, and so it is coming from where? Cells in the duodenum. So in the duodenum there is a CCK cell, there is a secretin cell, okay, and whereas the whereas the secretin cells are more sensitive to the pH. CCK cells in the duodenum, they are more sensitive to the amount of fats that is coming and amount of amino acids that is coming. Okay, they are sensitive, and in response to that, the CCK cells will secrete CCK. The CCK will act on the on this again 7 transmembrane G protein coupled system. It will act. It will release calcium ions, and that those calcium ions are responsible for what? Calcium ions are responsible. What do you think it will be doing? Calcium ions. What do you think they will be doing? Trafficking of the vesicles. What is it? Trafficking. You mobilize, okay, there will be cytoskeletal proteins, okay, they will be mobilized, and the zymogen granule will now merge with the plasma membrane. It will release its content, it will go into the primary duct, okay, and it will start moving along the duct, and as you go along the duct, let us see what happens to the. So what do we have here? So we have these are hundreds and hundreds of acini. These are the hundreds of acini. They will have the duct. Here is a larger duct here. And if you take the lining of a duct, you just say so here we have done this 100 times. We are looking at a single cell of what? The duct. And the moment you see a duct, we immediately should ask one thing, which is the basal side, which is the apical side, and in the basal side you must have the enzyme system. What do you call it as? Sodium potassium ATPase. So do you see a sodium potassium ATPase here? So this is the capillary, this is the basal side, and that's the lumen. Okay, and on the basal side you have you have the pump, and the pump keeps on pumping the sodium to the outside. As a result of you have too much sodium, as a result of you have this another system which is the antipode system, and what is that antipode system? Is driving the H ions away from the lumen towards the blood. Do exactly what you did opposite to what you did in the stomach. In the stomach you were driving the bicarbonate ion towards the capillary and the H ions towards the lumen. Now you will do the opposite. What would you do? You would drive the H ions here and the bicarbonate ions are coming from the story. The carbon dioxide will go combine with water, carbonic anhydrase, bicarbonate ions are going there with the help of an antipode system which is exchanging chloride ions to bicarbonate ions. You need to recirculate the chloride ions repeatedly because it's only the antipode system. So the chloride ions keep on circulating and they facilitate the transfer of bicarbonate ions into the lumen. The system is so efficient that the bicarbonate ions in the lumen are about 5 times more than in the blood. Are you understanding? You see, one way to look at the biology is that biological systems are capable of continuously partitioning things. Are you with me? Partition. So in this case, what is being partitioned? Bicarbonate ions are being partitioned. They are being driven where? Into the lumen. Okay. What is the purpose? You have to neutralize. Neutralize what? Acid that is coming from the center. Coming from where? Stomach. Good. Okay. So you have the. Are we okay with this diagram or not? Is this, yeah, go ahead.
How do these cells get enough CO2 to produce enough bicarbonate?
The problem is CO2 flows from anywhere to anywhere because of extremely high solubility. CO2 can go anywhere, and every cell, every second is generating carbon dioxide. So we have no dearth of. Okay, even if you say that this particular cell may not generate enough this thing, it will flow. The partial pressure of and carbon dioxide is not stopped anywhere. For carbon dioxide, there is no compartment. Okay, all right. So we have once the bicarbonate goes, the sodium ions go by the transcellular pathway. Yes, please.
So in the case of parietal cells, secretion of H plus caused alkaline tide in the blood.
Yes.
Does this later secretion of
It no. It doesn't cause. Does it really change the pH? That's your question. The pH doesn't really change. There are enough buffers. There are enough buffers in the blood to absorb those ions, so that you take the ions but don't literally. Even in the case of bicarbonate, there is a very slight change may be there, but there is no major change because the blood has enough buffers. Okay, now comes, you see, I mentioned a particular term which did not click, but I will reuse the term again and I will give its meaning because it is very interesting. I will tell you what. The chloride ion channels. What did I say? Chloride ion channels. Okay, they broadly form into. So there is a channel. It is a channel. Okay, means what? Means what? It is a system of proteins. Where does it sit? In the plasma membrane. Okay, you can. It is closed. Okay, but if you have sufficient amount of voltage difference across the plasma membrane, it can open. Therefore, we will call it as voltage gated chloride ion channels. We have done it 100 times, chloride gated. Okay. Now, there are different families. Chloride ion channels are again dozens of them. Here we are talking of one particular type of chloride ion channel. So, what is this? What is this? This is the same image as we have seen in the previous, same image. Little difference, I will tell you. So, what are we talking about? We are talking about the lumen of the duodenum. On the basal side, there is a capillary somewhere. Where is the pump? The pump is somewhere here. There is water. There is carbon dioxide, carbonic acid, bicarbonate, and then it is an antipode system. Bicarbonate goes here, chloride comes, and chloride keeps on circulating. Now comes a problem. There is a human genetic disorder in which this particular chloride ion channel, okay, which allows the chloride ion to circulate. You see, every time a chloride ion circulate, that circulation is necessary so that this antipode system has to work. And its antipode systems are 1 for 1, 1 for 1. What do you have? 1 for 1. Every time it goes through a cycle, it does not use energy. It does not use directly, it is not a pump, but it allows what to go? It allows the bicarbonate ions to go in this direction and the chloride ions to go in another direction. But the chloride, you need the supply of chloride so that the cycle can continue, and for providing that you have this channel. This channel is a protein. That protein comes from mRNA and gene. In humans, that gene is known to have mutation. Okay, and the persons who have that mutation are known to suffer from a disease called as cystic fibrosis. What is the disease called as? Cystic fibrosis. Say that again. In the cystic fibrosis, what happens? Because the system does not work, they have a lot of accumulation of mucus. Okay, this system also works in our lungs and in the pancreas. In both the places, there is accumulation of mucus, and that can give rise to many serious problems. Okay. Therefore, this channel is called as cystic fibrosis. I want you to read here: cystic fibrosis transmembrane conductance regulator. Very funny name. Abbreviated as what? CFTR. CFTR. What does CFTR stand for? So, what is CFTR? CFTR is, if you are, most of us, we are healthy. We are healthy because there is no mutation and we are doing fine, and that is we are fine because the chloride ions are just going through this channel. Yeah, through this channel. It is permitting it's permitting chloride ions to continuously leak out. Now I am talking about patient. What is the problem there? There is a mutation in the gene. As a result of that, that protein is defective. As a result of that, it does not allow the chloride ions to go out. Are you with me now? So the whole system is now screwed up. Are you okay so far? Okay, and then the patient suffers from. Okay, so just remember as to what is this CFTR. Is every chloride channel a CFTR? No. There are several of them. Only one family is a chloride channel. Okay, so we are talking about look at that image, on very nice image. This is histological image, now actually immunocytical image, in which you have used antibodies against which hormone, which peptide? Secretin. Okay, it is about 27-28 amino acids, small peptide. You are able to. So okay. Can you see this brown dot here? What does each brown dot represent? Secretin, secreting cell. Okay. All right. Good. So here we have. So let's understand this image. This is a very interesting image. The food has been delivered in the form of chyme from the stomach in the duodenum. And look at this arrow. And en route, it stimulates the same thing. It stimulates what? And what? CCK secreting cells. Now, whereas the secretin goes and stimulates the cells of the duct so that they release what? Bicarbonate and water, and the CCK will stimulate the pancreatic acini cell. So therefore, the cells will start discharging more and more of enzyme. So there are two components. Pancreas has you have to release a lot of synthesize enzymes from the zymogen granules. Okay, okay so far. And then in the duct you have to give bicarbonate ions, okay, and that is promoted by the CCK. Good. And you please read this, and this will give you the basic information as to how the signaling happens in the very interesting graph. We are looking at the duodenal pH on the x-axis. Okay, so far, and this is what strong acid. Hello, are you with me? And little weak acid, and a weak acid. So we are monitoring the pH of the duodenum, number 1, and at the same time we are trying to correlate it with the bicarbonate ion in pancreatic bicarbonate output, how much of it is giving. And what is the correlation when the pH is what? Very high. How much is the bicarbonate concentration? So much. As you make it less and less and less acidic, which means your pH is going to 4 and 5, what happens to the bicarbonate? A very beautiful correlation. Okay, the more the acidity, the more the bicarbonate. I mean, that's common sense. Okay, and but that has been scientifically proved here by establishing the correlation between the release of. Okay, I'll stop now about the importance of description of pancreas. You will read more about it in the slides. I'll move on to yet another interesting organ, that is liver. And I'm going to talk another 10 minutes. I'm going to talk about liver. Yes, liver is a very interesting organ. It's very large, more than 1 kilo. One of the largest organs, one of the most important organs, although there is nothing endocrine about it, but it's a laboratory, hundreds of things. It's a storehouse for glycogen, it's storehouse for lipids, it's an organ for the synthesis of a large number of proteins, the proteins which are responsible for the coagulation of blood. N number of proteins are being actually, it's huge. So let's see what is this liver. Now focus on what we are talking about, liver. I am sure you can see the liver there, I am sure you can see the gallbladder there. Hello, are you okay? Can you see the gallbladder there? Okay. Then of course you can see the rest of the structures. Now follow this. Be with me, this is interesting. Blood is drawn to the liver through 2 independent sources. How many? 2 independent sources. One is very simple. One is dorsal aorta, heart, aortic arch, dorsal aorta gives branches. One of them is called celiac artery, again branches, and one of them becomes hepatic artery. What does it do? It takes blood to the heart. I am sorry, made a mistake. Blood to what? Blood to liver. Very good. And it is oxygenated blood. So it's carrying. So the oxygen supply to the liver is happening via this supply. Hepatic artery. Done. Everybody, you are good so far? Good. The second supply is, that's even, yeah. The second is, you have, okay, can you see the, this is we have removed lot of alimentary canal, but lot of is still there. So you have, this is the intestine, and this is the ascending colon, horizontal colon that is broken, and this is the descending colon. The blood which has been, the arterial blood which has been taken to the alimentary canal, follow this. It has given away oxygen to the intestine. Are you okay? Then that blood has again got together in the venous system. Okay. Does it have oxygen? No, it is deoxygenated, very low, whatever, it's low. Now that should normally go directly to the heart. It's deoxygenated blood. It should go to, it's deoxygenated. So arteries went to the intestine, they gave away their oxygen, that blood got into the venous system, it should go to the heart. It doesn't. So the bluish vessel here, which indicate the veins, which are bringing the deoxygenated blood from the entire intestine, it goes to the liver. So this is what, this is the second blood supply to the liver. Are you with me? I said how many liver receives blood supply from 2 sources. One was hepatic artery. This blood supply is called as hepatic portal system. What do you call it as? Hepatic, say that again. Don't confuse. So whereas the liver will get its supply of oxygen through the hepatic artery, the liver will get all its food, which not that liver needs it. No, liver needs to process it. Okay, and for processing that food, we have the nature has provided with the portal system, which does not take oxygen to the liver, but which takes the all the digested food material from the intestine. Now, there is again additional twist in the tail. Let us go to the intestine. What it has absorbed? Okay, it has absorbed the proteins that you have taken. They have been broken down. Finally, amino acids. The carbohydrates now, let us presume they are reduced to fructose, glucose, galactose, etc. And fats are there. Fats are also digested. About that, I'll listen to this. So, three forms: digested fats, proteins, carbohydrates. Whereas fats get into the lacteal system, we talked about it, okay, and then it is taken. Okay, the broken down and digested carbohydrates and proteins, which is amino acids, okay, they are taken by this system to the liver. Are you with me? So, it is not, I cannot totally say that everything that is absorbed goes to the liver. No, no, the digested carbohydrates, okay, so far, which means glucose, etc., small monomers, okay, and then the amino acids. They will go where? Absorbed at the level of intestine. They will go they will be taken where? Liver. How? Hepatic. Say that loudly. Hepatic portal system. Okay, good. Now remember this, and in the villi, of course, there is a lacteal system, and the lacteal system will carry the fats. We have a different system for that. So liver, actually, we have seen this in the slide. Liver actually is made up of what you call as tiny lobules. Now, what is this a lobule? I will tell you what. This is okay, you take a cylinder, okay. The diameter of a cylinder is less than 1 mm, 0.8 mm or, okay. It is not a perfect circle, but it has angles. Angle is something like this, maybe 5 or 6 angles are there, maybe like a structure of benzene, 6. It is a cylinder like this. Is it really a cylinder? No, I am still calling it a cylinder. Are you with me? Is it a circle there if I take a transverse section? Yes or no? Is that a, okay, I will take you to the next slide, that will clear the aspect. Is that a circle? Not in strict sense, but something like this, okay. Now, but it is like this, and then as you, this is length maybe about 4, 5, 6 mm, but then it tapers. So, what am I doing? I am giving you a rough idea of what a lobule is, and there are thousands of such lobules in the liver, okay. Now, we have to understand the structure of a lobule, okay, which is a unit, okay, lobule, one unit, and there are thousands of them. If you have understood the structure of one lobule, you have understood the structure of all the lobules, but understanding the structure of one lobule is very important, and that is what I am going to do. So, what I am going to do is, I am going to talk to you about a single lobule. I talked about this, and I am going to talk it. So, we are looking at a single lobule here. So, can you see there is 1, 2, 3, about there are almost 6 faces are there and 6 angles are there, yes or no? Good. Now, at every corner there are 1, 2, 3, 4, 5, 6. How many corners are there? 6 corners. If I take a transverse section of a lobule right in the middle, I am going to get 6 corners, 6 angles. At each angle I am going to get how many tubes? Can you count for me how many tubes are there at each corner? 3 of them. People also call them as a triad. Okay, so far? Very good, very clear. Now, out of the 3 tubes, there is one red here. Red means it must be, it's for blood. It must be carrying what kind of blood? Oxygen. It must be branch of what? Hepatic artery, dorsal aorta, celiac artery, hepatic artery, and a branch here, and then thousands and thousands and thousands, and so one is here, one can you see red here. So the ultimate branch of that goes at all the 6 corners, one of the 3. Are you done? The second one is the blue one here, the blue here at the corners. Don't go to the middle yet. I am not ready for that. There is a blue one there. Now the blue one is coming from where? Hepatic portal system. So the hepatic portal system now is the blue one. This blue one here is a part of hepatic portal one. Okay. Now at the center of each lobule there is an opening, and that is called a central vein. What do you call it as? Central vein. Now, just as in a bicycle, you know, we have a central hub and spokes. Okay. In the similar manner, I am sorry, I am exceeding my limits. We will stop now.